is about what's happening in terms of the transfer of these gases at the level of a single cell. Really, how do cells use glucose, which is their main source of energy, to make ATP, which is our body's main source of energy currency, of how we can use energy or harness that energy to do cellular work, right? To do things in the cell. And so we're really going to delve into how the body takes the food that we eat on a cellular level and turns that into energy, okay? And so objective 1, to understand how our bodies use biochemicals to harness chemical energy. Okay, let's start with a few teaser questions. So I'm going to relaunch the poll here. Okay, let's take a look. All right, so a lot of response for C. Energy is released when proteins are broken down into amino acids. And so the reaction is exothermic and catabolic, right? So that's correct. Exothermic meaning that energy is liberated. Catabolic means that larger molecules are broken down. And one way that I like to kind of remember this, it's a kind of silly way, but I look at the X in exothermic and the T in catabolic, and I think about the positive energy that is being created or liberated as we break these molecules down, okay? And so that's going to be synonymous with breaking down our biomolecules to help liberate some of that energy. On the other hand, endothermic reactions or anabolic reactions are building up. So it's turning glucose into glycogen or amino acids into proteins. And so we're storing up the energy from the reactants, which are, glucose or amino acids, into the products, which then become glycogen or proteins, as just as examples. Okay. So let's go ahead and start here. Oops, yeah, let's skip over this. So we're looking at ATP here to start. And ATP, I kind of talked about it earlier. It's actually made from one of our nucleotides, if you remember adenosine, and then we add 3 phosphate groups to that molecule. And so this is our essentially an energy exchange, an energy currency, if you will. And so within that third phosphate, right, we've got ADP, and when we add a third phosphate there, there's a lot of energy stored in that third bond. That energy is energy that we can liberate to do cellular work. When we talk about cellular work, we mean things like making proteins, making hormones, making receptors, moving things in and out of the cell. That type of cellular work requires energy, and ATP is the energy format, or the energy currency, as I like to call it, that we use to do that cellular work. And so I like to think about ATP as a battery pack. I don't know if any of you have like a mobile, temporary backup charge for your battery that you can take with you, but it's kind of like that. The only reason that you charge that thing is that so that you can have it when you need it, to use it when you're outside and you're not accessible to a tap. And ATP has a similar role. The only reason that we make ATP is really to break it, to use that energy to do cellular work, okay? So it really is just an energy reserve or an energy currency that we can then expend or use throughout our cells to achieve some of the processes that we need for survival. And so if we look at the way that we make ATP or ATP synthesis, we've got that ADP, we add that third phosphate, and that allows a lot of energy to be stored within that molecule. And so we then produce ATP and then some water as a byproduct. And so the change in energy here is about 7 kilocalories per mole, meaning that this is an endergonic reaction, or we are storing up or building up energy within this molecule. And so this is also going to be a condensation reaction, hence the production of water as the byproduct. Okay, we talked about condensation reactions before as well. Now, there are two ways or two mechanisms to make ATP. In other words, to add that third phosphate to ADP, we can do so via substrate level phosphorylation. And so in substrate level phosphorylation, we have another molecule in the cell that has a phosphate on it. And through enzymatic reactions, we simply take that phosphate from molecule X, add it on to ADP, and then at the end of this reaction, we get molecule X on its own without the phosphate, and then ATP. So essentially, we steal a phosphate or borrow a phosphate, if you will, from another molecule that has a phosphate on it. And that is called substrate level phosphorylation, okay? This can happen in the absence of oxygen, which is a really important distinction to make between the other mechanism that we're going to talk about, which is oxidative phosphorylation. And oxidative phosphorylation requires energy. That's the first thing you want to make a note of, hence the term oxidative. So this cannot happen independent or in the absence of oxygen. It's also going to require the electron transport chain, which is a series of electrons embedded in the membrane of our inner mitochondria, so very specific part of the cell, the mitochondria. Within the membrane inside that mitochondria, there are a bunch of electrons in that membrane. And we'll talk about this in more detail, but essentially via oxygen and the passage of electrons across this chain, we then add an inorganic phosphate, meaning a free-floating phosphate not attached to any other molecule, to our ADP, and we can make ATP that way, okay? So a very simplistic way of looking at both of these is that substrate level phosphorylation happens independent of oxygen, happens by grabbing or stealing or borrowing a phosphate that's attached to another molecule, whereas oxidative phosphorylation happens inside the mitochondria specifically, requires oxygen, and uses a free-floating phosphate that's not attached to any other molecule, okay? Let's look at how ATP is broken down. So I mentioned earlier that ATP is only made to be broken, right? Just like the way you only charge your backup battery so that you can use it and run it down later on. And so when we break down ATP, we release that temporary energy that's stored in that third phosphate. And so we're going to have to insert water here. This is a hydrolysis reaction. So we insert water, and then we end up with ADP, like just the way we started, and then that phosphate, and then the energy, really importantly, the energy that we're trying to liberate to do things within the cell, okay? So again, this is a hydrolysis reaction, meaning that we have to insert water in order to break that covalent bond. Okay, now we're going to speak about what's called glucose oxidation. This is aerobic respiration. And aerobic meaning that oxygen is going to be required for the full completion of this process. And essentially, we're going to be speaking about it in the context of glucose, because glucose is our main energy reservoir. We talked about this. Our cells prefer energy coming from glucose, which is coming from carbohydrates. And so we're going to speak about it in the context of breaking down one molecule of glucose and liberating the energy to be used within our cells. But I want you to also be thinking that we can also break down or oxidize other biomolecules as well. We can oxidize fats, which we'll talk about later on, and we can also oxidize proteins. But in order to explain cellular respiration, we're going to use the context of a single molecule of glucose, okay? Because it's the most common, it's the most abundant, it's the favorable energy currency for our body, for our cells. And so the central reaction of energy metabolism of glucose oxidation is C6H12O6, which is essentially glucose, plus oxygen, is broken down to 6 CO2, 6 water, and energy. Okay, this is how we express that breakdown of glucose to liberate energy. And so we should theoretically liberate or get from this breakdown of glucose about 686 kilocalories per mole. Okay, that's the energy change that's happening here.
So when we're breaking down glucose, are we always adding energy?
So we're
Are
not,
we taking it?
sorry, say again.
Are we taking or adding energy?
So we're liberating the energy that's stored here within that glucose.
Okay.
So we're going to take glucose and oxygen, and we're going to liberate CO2 as a byproduct, water as a byproduct. Remember, this is breaking, so that's a condensation reaction, and then energy that we want to liberate from that glucose, okay? Okay. I think I was going to say that we also can express this reaction in the reverse. So if you think about plants, plants have the ability to photosynthesize, so they can take energy from the sun, water, and they can use CO2, right? Plants need CO2, and they can release oxygen and glucose, right? So we can think about this in our physiology happening in the forward direction, but in terms of plants, this happens in the reverse reaction as well, right? They take the sun's energy, water, CO2, and they give us glucose and oxygen. This is why plants and humans, and not just humans, but other species as well, work in this symbiotic relationship where they produce what we need and we produce what they need on the other end, okay? Here we can illustrate how that ATP is made from the liberation of the energy that's stored within that glucose. So here's our initial reaction, right? That glucose, C6H12O6, plus some oxygen, and that is turned into CO2 and water. And then more importantly, the most important part of breaking down that glucose is liberating the energy that's stored inside. And so when that energy is liberated, that's how we make ATP. We take that energy and we use that energy to add a third phosphate onto ADP. And so trapped within that ATP molecule is the energy that we liberated from that glucose. That's what cellular respiration really is. Taking the energy harnessed from the food we eat, most importantly, glucose as our main example, using that energy to phosphorylate ATP, to use it as our energy currency. And then we only make ATP, as I mentioned before, in order to break that ATP, liberate that energy so that we can do cellular work. And again, cellular work here is things like cellular movement, right? Moving things around in the cell, making proteins, making hormones, receptors, etc. Recycling, getting rid of waste or trash material in the cell as well. Also making molecules, and then transporting things across the membrane. So those are some examples of what we mean when we say cellular work, and that requires the energy that we have stored in ATP that we actually liberated from the food we eat, in other words, glucose, okay? Let me pause here and see how we're doing with that so far, and we'll keep going. So any questions on that so far?
Can you explain a little bit more what you mean by liberating?
Excellent, yeah. So liberating just means that we're harnessing the energy. We're taking the energy. This is an energy transfer reaction. Okay, so the same energy, if I jump back here, the same energy that plants use from the sun to store into that glucose, we can liberate it and turn it into what we need, which is ATP here. And then that ATP can be used to do cellular work. So when I say liberating energy, I'm just meaning an energy transfer, right? So taking the energy from glucose, putting it into ATP to make ATP, phosphorylate ATP, and then having that ATP as our energy currency in the cell to help with the processes that we need inside our cells, okay? All right. Okay, so here is a more detailed reaction of that glucose oxidation. So again, we have glucose, we have oxygen, and we should be able to phosphorylate. We should have 38 ADP. And if we have 38 phosphates, then we can phosphorylate and make 38 ATP on the other end, and we'll release some water and we'll release some carbon dioxide. So a simpler way to say this is that with the energy that is liberated or released from one molecule of glucose, which we have here, we should have enough energy in that glucose to phosphorylate 38 ADP, in other words, to make 38 ATP, okay? Theoretically, in terms of the energy movement inside of that glucose, okay? And so what we do see in our physiology is slightly different, and I'll explain why in a little bit here. But in our physiology, when we break down 30 ATP, when we break down 38 ATP molecules, we only have 266 kilocalories worth of energy there. Okay, let me explain that in another way. So if we took the 38 ATP that we made from this one molecule of glucose and we used that in our cells and we broke that down, we would only have 266 kilocalories to use in our cells to do work. And so what that exposes is that there's a gap, okay? Because we know from our overall reaction of glucose, if we go back to this slide, there should be 686 kilocalories worth of energy inside a single molecule of glucose. But in terms of what actually happens in our body, we only are able to use 266 kilocalories of that energy. And so that gives us a gap of 420 kilocalories. So there's a difference of 420 kilocalories. And what this explains to us is that this is not a perfect process. A lot of the energy that's taken from the food that we eat and turned into ATP, in the process of making ATP, we lose most of that energy. We lose most of that energy as heat, okay? Jumping back to this slide, and I apologize for kind of skipping all over the place, but on this slide, we can see that, right? Most of the energy that's taken from the glucose doesn't actually make it into ATP. Most of it is lost as heat, okay? So it's kind of the price that we pay, the energy expenditure, because we are not perfect beings, and because this doesn't happen in a perfect way, a lot of this is just lost as body heat, okay? Same thing when we liberate that energy that's stored in ATP to do work. A lot of that is simply lost as heat as well. And so coming back here and explaining that in the context of the numbers here again, we should have 686 kilocalories of energy in one molecule of glucose. But in actuality, in our physiology, when we break down one molecule of glucose, which is 38 ATP, we only liberate 266 kilocalories in terms of what actually happens in our physiology. So this is a theoretical value, and this is our actual physiological value. And so that exposes a gap of 420 kilocalories, which tells us that this has been lost as heat, which is the price that we pay for transferring that energy to make ATP. Okay, so that tells us that this is a 38% efficient process. In other words, only 38% of what we liberate from glucose we can actually harness in our cells to do work. And so 61% of that is simply lost as body heat. All righty.